Welcome back, everybody. Ready to dive into something pretty cool. We're going to take a close look at these thing called glycophorins.
Glycophorins.
Yeah, so they're these proteins. They live on the surface of your red blood cells.
Right.
And they're like super important for figuring out like what your blood type is.
Yep.
Like if you're ABO, all that stuff.
Mm-hmm.
But they're also like really crucial for how your immune system knows which cells are yours and which ones aren't.
Exactly. They help your body tell friend from foe, essentially.
So for this deep dive, we're going to crack open chapter 5 of this research paper. And it's all about how scientists are using computer models to like really study these glycophorins.
Yeah, they're focusing on 3 specific ones.
Okay, hit me with those.
Well, we've got glycophorin A.
GPA.
Exactly. Then there's glycophorin B.
GPB. I'm sensing a pattern here.
Right. And then there's this really interesting one called GP.mer.
GP-mer. Okay, so we've got our lineup. But what makes them so special? Why are we spending a whole deep dive on these guys?
Well, you see, even though they're all kind of related, these glycophorins, they each have like their own personality. Personality in a way, I mean, in terms of their structures. And those differences, they actually affect how they like behave and interact with other molecules in your body.
So it's not just a one-size-fits-all kind of thing with these proteins.
No, not at all.
Got it. So walk me through these personalities then. Let's start with GPA. What's its deal?
Okay, so GPA, this research paper, it describes GPA as being like super stable. And when it pairs up with another GPA, they form this like basket shape. We call that a homodimer.
Homodimer. So 2 GPAs just chilling in a basket.
Basically, yeah. And that basket shape, it's important because it creates this little pocket that can attract and hold on to other molecules, like a docking station.
So GPA is like the organized one, right? Always keeping things neat and tidy with its little basket.
You could say that. Now, GPB, on the other hand...
Uh-oh, here comes the troublemaker.
Kind of. GPB is a lot more free-flowing. It's got this kind of messy, flexible structure, which honestly makes it a pain to study.
How come?
Well, you see, the antigens, those molecular flags we talked about, they're kind of scattered all over GPB's surface.
Oh.
So it makes it hard for antibodies to, like, consistently recognize them. It's like trying to grab onto a fish that's constantly changing shape.
Slippery little guy.
Yeah.
And what about GP-mer, this hybrid one? What's its structure like?
Well, GP-mer is kind of cool because it seems to be a blend of the two. Like, it has some of that GPA stability, but also a bit of that GPB flexibility.
The best of both worlds, maybe.
Maybe. It definitely makes it a fascinating subject for research.
Now, you mentioned that scientists are using, like, computer models to get a good look at these structures, right? Can you break that down for me? Like, how does that even work?
Yeah. Imagine you could zoom in, way in on a glycophorin and see every single atom, every little twist and turn.
Wow.
That's basically what these computer models let us do. They create these incredibly detailed 3D images of the proteins.
So it's like a high-tech microscope.
Exactly. And these models, they can help us understand how the shape of a protein influences how it actually works in the body.
That's pretty amazing.
Yeah.
But, okay, how do we know that these computer images, like, actually match up to what's really going on inside us?
That's the million-dollar question. And that's where lab experiments come in. It's a two-pronged approach.
I'm 2 prongs, okay.
Yeah. So the computer models give us a hypothesis, a really specific prediction of what we might find. And then we take that prediction into the lab and use experiments to test it out in a real biological setting.
So it's a team effort. The computer gives us the blueprint, and the lab puts it to the test.
You got it.
Cool. So we've got these different glycophorins, each with its own unique structure. And we know they play a role in determining our blood types and helping our immune systems do their job. But can you explain a little more about what antigens actually are? Like, why are they so important?
Okay, so imagine your cells all have a unique barcode, right? That barcode tells your body, hey, this cell belongs here. It's one of us. And that barcode is made up of antigens. They're like tiny flags on the cell surface that the immune system uses to distinguish between what's you and what's not you.
Okay, I'm with you.
So when your immune system sees a cell with familiar antigens, it's like, all right, we're good here, move along. But if it encounters a cell with foreign antigens, it's like, red alert, intruder, time to fight.
Ah, so that's why blood transfusions have to be so carefully matched.
Exactly. If you get a transfusion with the wrong blood type, your immune system freaks out because it sees those unfamiliar antigens and tries to attack the new blood cells. Not good.
Yeah, no kidding. This is really fascinating. So
Yeah.
we've got these glycophorins, different structures. They've got these antigens that are key for blood typing and immunity. But do they all present their antigens in the same way? Or is there more to it?
That's a great question, and the answer is nope, they definitely don't all play by the same rules. We've already seen that GPA and GPB have pretty different structures. And guess what? Those differences actually affect how they show off their antigens.
Okay, spill the tea. How does GPA do it?
GPA, Mr. Organized over here, it presents its antigens in a very orderly way, thanks to that nice basket-like structure.
So it's like GPA is rolling out the red carpet for the immune system. Here are my antigens, nice and easy to see.
Exactly. Take the M antigen, for example. It's a really important one for determining blood type. And on GPA, it's like front and center, easy for antibodies to spot and recognize.
Makes sense.
Yeah.
And what about GPB? Does it follow the same etiquette?
GPB? Not so much. Remember, GPB is the wild child, always changing things up with its flexible structure. So its antigen presentation is a little more, shall we say, unconventional.
Unconventional, how so?
Well, the research highlights the N and GNU antigens on GPB, but because it's so flexible, those antigens aren't always in the same place. So it can be harder for antibodies to reliably recognize them.
So it's like GPB is playing a little game of hide and seek with the immune system.
You could say that.
And what about GPMER, our hybrid friend? How does it present its antigens?
Honestly, GPMER is a bit of an enigma. It has this unique mix of antigens, MIA, MER, U, and S, and how they're arranged on its surface likely affects how antibodies interact with it.
So it's like GPMER has its own secret language that the immune system is still trying to figure out.
Something like that.
Now, all of this is based on computer models,
Yeah.
right? So how do we know for sure that these findings hold up in the real world? Like, how do we bridge the gap between what we see on a computer screen and what's actually happening inside our bodies?
Well, that's where this whole idea of combining computer models with, like, real-world experiments, that's where that really comes in.
Right. So it's like a scientific tag team.
Yeah, exactly. The computer models are like our roadmap, you know? They give us these really detailed predictions of what we might find, and then we take those predictions and we go into the lab and we use experiments to actually see if those predictions hold up in a real biological system.
So it's a two-way street. The computer gives us the clues, and the lab helps us crack the case.
Exactly. And this kind of back and forth between the computer and the lab, that's how we make sure we're on the right track, scientifically speaking.
And that approach, it's been working out pretty well, huh?
Oh, yeah. We've had some really exciting breakthroughs. Like, remember how we talked about that interaction between GPA and the band 3 protein?
Yeah, that was pretty cool.
Well, scientists used computer models to pinpoint the exact amino acids where those two proteins were most likely to connect.
Whoa.
And then they used lab experiments to actually confirm that those specific amino acids were crucial for forming the right B antigen.
So the computer model was like the puzzle pieces, and the lab experiment snapped them all together.
Yeah, that's a great way to put it.
It's amazing to think that we can study these interactions at such a tiny level. It really makes you realize just how complex and awesome our bodies are.
Absolutely.
But let's zoom out for a second. What does all of this research actually mean for us in the real world? What's the big picture here?
Well, one of the most obvious applications is in the field of transfusion medicine.
Right. Blood transfusions.
Yeah. The more we understand about how these glycophorins work and how they present their antigens, the better we can match blood donors and recipients.
So we can avoid those bad reactions that can happen if you get the wrong blood type.
Exactly. But it goes even deeper than that. This research has huge implications for personalized medicine, too.
Personalized medicine, that sounds super futuristic.
It kind of is. Imagine a world where we could create treatments that are tailored to your specific blood group profile.
So instead of just giving everyone the same treatment, we could design therapies that are specific to your individual blood type.
Exactly. And that could mean treatments that are way more effective and have way fewer side effects.
Wow, that would be a game changer.
It really would. It's one of the most exciting frontiers in medicine, I think.
Well, I'm sold. But I gotta ask, what are some of the challenges that researchers are facing when they study these glycophorins? Like, what are some of the big questions that still need answers?
Well, one of the biggest challenges is trying to understand how these glycophorins interact with antibodies in the, messy environment of a living, breathing human body.
Yeah, I bet that's not easy.
It's not. I mean, we have these awesome computer models that give us a starting point, but then we have to confirm those predictions with experiments that actually mimic real-life conditions.
So it's kind of like taking those computer models for a spin in the real world.
Yeah, exactly. And just like in the real world, sometimes things don't go exactly according to plan.
I feel that.
Yeah.
Are there also, limitations to what we can do with these computer models and experiments?
Of course. Like with any technology, there are limitations. With computer models, we're always trying to develop better algorithms, to capture all the nuances of these complex biological systems. And then with lab experiments, well, it's tough to perfectly recreate the dynamic environment of the human body in a controlled setting.
Right. That makes sense. So it's like this constant dance between like predicting and confirming, always pushing for a better understanding.
Yeah, pretty much.
Well, even with those challenges, this research is obviously doing some amazing things. What are some other areas of research in this whole glycophorin world that you're excited about?
Well, one area that I find really fascinating is this idea of using glycophorins as targets for drug delivery.
Targets for drug delivery. What do you mean?
Like, imagine if we could design drugs that specifically bind to those glycophorins on the surface of red blood cells.
Okay.
That way, we could deliver medication directly to the cells where it's needed most.
Wow. So it'd be like a GPS system for medicine. You can make sure the drug goes exactly where it needs to go.
Exactly. And that could make treatments way more effective and reduce side effects.
I can see why you're excited about that.
And then there's this other possibility of using glycophorins as biomarkers for disease.
Biomarkers.
Yeah, like
Right.
if we could analyze the way these glycophorins are expressed, or if we could spot any modifications to them, we might be able to detect certain diseases earlier, before they become a big problem.
So those tiny proteins could help us catch diseases early on.
Potentially, yeah. And that can make a huge difference in terms of treatment and outcomes for patients.
It's mind-blowing to think that these little proteins on our red blood cells could have such a big impact on our health.
It really is. And honestly, we're just scratching the surface when it comes to understanding these glycophorins and all the different blood groups they represent.
There's so much more to learn. It's pretty exciting.
Definitely.
It really makes you appreciate, all the hard work that scientists do. Exploring these tiny little things can lead to some pretty big discoveries.
Absolutely. And one area that's really caught the attention of researchers is this idea of engineering artificial blood.
Wait, artificial blood, is that even possible?
Well, it sounds like science fiction, right?
Yeah.
But it's a real area of research and it has huge potential. Imagine a world where we could just create blood in a lab.
Oh.
And we could tailor it to meet specific needs. No more worrying about donor shortages or compatibility issues.
That would be incredible, especially for emergency situations or for people with rare blood types. It'd be a total game changer.
Totally.
So as we wrap up our deep dive into this glycophorin world, what's the main takeaway you want our listeners to remember?
I think the biggest thing is to realize that these glycophorins, they're not just some simple little tags on our red blood cells.
Right.
They're actually really complex and dynamic molecules. They've got this fascinating evolutionary history, and their future is even more exciting.
We've seen how their structures, how they present those antigens, all of that. It affects so much. Blood transfusions, personalized medicine, it's all connected.
And as we keep learning more, combining computer models with those lab experiments, who knows what else we'll discover?
Yeah, it feels like we're just getting started.
Exactly.
It's a good reminder that science is all about exploring, and that even the smallest things can have a huge impact. Maybe one day, thanks to these little glycophorins, we'll have even more breakthroughs in healthcare.
I'm definitely looking forward to it.
Me too. Well, I think that about wraps up our deep dive. A huge thank you to our expert for taking us on this journey.
Happy to be here.
And to all our listeners, thanks for tuning in. Keep those brains buzzing, and we'll be back soon with another deep dive into a brand new topic.